Hello, I am Sunny Zhou from Northeastern University in Boston. Today, I will talk about characterization of process-related impurities. Because unfortunately I won't able to meet you in person and chat about all the program in my laboratory, I decided to include a few more topics than I normally would do for a short talk. So I will go pretty fast, but I will be happy to share more details and also some ongoing program with you offline. So today, I will talk to you first about impurities, especially how we define impurities. One thing people always consider as impurity are whole cell proteins and other excipients. But I think other impurities people talk about is product-related variants. In generally, people think about impurities as something bad or inactive. I want to use this talk to illustrate some other thought about impurity in terms of their function and impacts. And in terms of impact, this is a long-going program in this field about critical attributes and also programs about quality by design. Again, this is ties into impurity into different tiers, what are important, that's related to in vivo functions as well. So there are several things I will share with you today. First part is about process-related impurities. I give you a few examples of upstream formulation and also some underappreciated factors such as light and cross-linking. And I'll very briefly talk about changes during the process. And then I will talk about characterization focus on especially analytical artifacts. I use deamidation and AAV as an example. Again, I will touch upon some new modalities, including gene therapy, adeno-associated virus such as AAV, and antibody drug conjugate. So my talk will try to mix these angles together so you will see how these different angles work together. So the first example actually is from Greg Flynn, who used to be a scientist at Amgen. Greg did a series of excellent work, look at various PTMs are endogenous to human antibodies. On this slide shows a particular one is deamidation, as you all familiar with. There's a couple of things to note. So this goes to the question, what is impurity? Do we consider endogenous existing PTMs as impurity or something natural to the antibody platform in general? Couple of things to note here. Antibodies is long-lasting drugs. The half-life in vivo typically around 2 to 3 weeks. So what Greg found out that he can mimic the various events in vitro and in vivo, and they are similar in parallel. So the in vivo data can support match to the in vitro data. So what he also found that the antibodies IgG recover from human serums, there's about 23% of these endogenous antibodies are deamidate. So again, this is a question, what are impurity, whether this deamidate variants consider impurity or endogenous variants or PTMs. And so one of the things to note is we typically formulate antibody around pH 5, and but in vivo application, the pH will be 7.4 and 37 degrees over a couple of days. So this also raises the question about how we control impurity and ties to the ultimate in vivo application. In other words, if you can control the impurity, in this case deamidate product, very well in formulation condition, but once we inject into a human subject, they deamidate very fast. And then how we consider which one is important, formulation stability or in vivo stability. Greg Flynn also did some excellent work on other PTMs, glycation, C-terminal-lysine, pyroglutamic acid, and also including covalent dimerization. We will talk more later on in this talk. So the first part of the talk is about upstream process. So what I call happier cells and a better product. Goes back a program maybe more than a decade ago. That time people really focus on to increase titer. What I want to show here is the reactive metabolized, which change alters during cell culture condition can have a direct impact of the composition of the product. And so this includes reactive metabolites and light and other things. So the first example is a previous unknown metabolite, methylglycol, that react with antibody product. And this is discovered during different cell culture conditions, particularly during chemically defined medium. And then we see a new peak, acidic peak over here. And there shows increase of mass of 54 and 72 Dalton. And this mass change for antibody wasn't in the database. Without going into the detail, we are able to figure out that this metabolite is methylglycol, is a dicarbonyl compound here, shows over here, that can react with arginine, form this 5-member ring adduct. Actually, it's well known methylglycol is a reactive metabolite during cell culture, and in fact, back in 1998, was reported in a CHO cell. What is relevant here is the cell culture change make methylglycol MGO elevate under this condition, resulting modification of this antibody. The other example is follow the same line during the cell culture engineering. AbbVie team added ascorbic acid, vitamin C, to the cell culture because vitamin C is commonly used as reductant to fight against the oxidative stress. In this case, the oxidation product of vitamin C shows in the upper right corner is become reactive. Okay, after carboxylation form a dicarbonyl compound, and that in turn react with antibody. And this is a report on the adalimumab humiria composition. So this shows that cell culture condition can process related product impurity variants. So the other example I want to share with you now is other factors which previously underappreciated perhaps. One thing is about light. So this is a program work with Ming Liu and colleagues at Amgen. So a little bit hard for you to see, but if you actually look closely, this is the anti-product. Without stress, there's some covalent cross-linking, and this is not disulfide bond because it cannot disappear at the reduction condition. And if we expose this antibody to other base and a light stress condition, there are more cross-linked species formed. And we have no idea of the mass change because they're too big at that time. And so we have to develop a specific workflow to look at these unknown cross-linking events. Without going into the detail, mass spec just not able to by itself solve this chemistry because for the most workflow, we need a predefined chemistry, mass, and site to identify the modification. So the paper talk about the approach we use, summarized here. So for all these previously unknown or not predefined modification, we have to develop a pan-specific chemistry for the target chemistry. Pan-specific method for the target chemistry. In this case, we label the protein with O18 water during digestion, and for cross-linked peptide, because they have two C-termini, compared to linear peptides only have one C-termini, and we're able to distinguish by the mass shift from O16 to O18 water. And at the bottom trace shows that some of the peaks was able to detect by all these peaks are detected by isotope labeling, but some of the peaks were missed if we only use the typical criteria to look at cross-linking events because some of them are miscleaved peptide. Again, without going into detail, we're able to figure out what the chemistry for this cross-linking, and it involves a oxidation of a histidine, and then cross-link another histidine with this oxidized species, generate a final cross-link product. And this, again, this is the first time ever discovered in a protein. Again, this is previous unknown. But once these unknown events become known, we're able to discover other similar cross-linking involve histidine. Shows here is an example from another company, I think it's Biogen. And this type of modification has also been discovered in biological system. And this light-induced cross-linking also lead to an industry-wide initiative in terms of safe light. So during manufacturing and storage, all the reactions vessel are exposed to UV light. And so there's because of this discovery, people are switching to safe light during the manufacturer environment. The other example is during small scale production, some of the reaction vectors actually exposed to light, so make the process less reproducible. So another topic I want to share with you, but I only mention very briefly, is some of the changes of impurity during the process. Because at the different steps, there's different conditions applied. For example, viral clearance involve low pH conditions. So that actually will change some of the impurity composition. Some impurity may disappear, some may appear. One example actually is zymogen. It's an inactive form of enzyme. This is particularly relevant to whole cell proteins. Again, I will not mention very much detail today. Feel free to talk to me about this ongoing program. Just give you a very quick example. There are lots of whole cell protein, and the detection of whole cell protein are still challenging, as you know. Some because they are low abundance, but sometimes also due to the fact that many of the analysis is based on mass spec ID or immunoassay, but not based on the activity assay. So one of the program we are doing is to using activity-based probe to function analyze these proteins. So now I'm going to move to the second part of the talk is about analytical artifacts. And this is, I think, underappreciated phenomenon, especially associated with characterization of impurities. So we use the example deamidation to illustrate some of the point of analytical artifacts. Shows here is a transformation of deamidation or summarization of aspartic acid to isoaspartic acid, as you know. What I want to emphasize here, this is a spontaneous process. And so in a peptide structure without a secondary structure, there's NG sequence deamidate quite fast with half-life about 1 day under physiological conditions. And asparagine followed by serine and histidine deamidate about 10 days. This sounds quite a long time, but I will show you even during analysis, this is quite fast events, may cause analytical artifacts. So this is deamidation is well-known antibodies, as illustrated here in the review. And recently, there's a new modality of AAV, and there's a highlight in Nature Biotechnology about a report from Jim Wilson's group about the deamidation of AAV particles. And Jim also highlighted in his paper, they actually did quite a thorough analysis, tried to tease out the issues, and are independent of vector recovery and conditions. I was able to talk to Jim about some of the complications, and I will be highlighting the next several slides. So here is a summary of their data. What I want to draw your attention to is the right panel. You can see during the analysis, they heat the sample to 90 degrees for 10 days. Elevated temperature will induce deamidation for the sample. And also the digestion is 37 degrees overnight. Again, this is quite a long time, can induce deamidation artifacts during the analytical process. So this shows the trend for deamidation. On the left side shows the sequence trend. And on the right side illustrates the point that if I have a protein is folded, shows in the spaghetti, but if once you denature that or digest into peptide, there's no secondary tertiary structure, this deamidation events happens typically much faster, illustrated by this red star and the boat, right? So this is the deamidation during analytical process. So that's artifact. So here, summarize the issue. So in the middle is the observation someone can have is contain asparagine and the diamide species that only shows aspartic acid, not isoaspartic acid species. But this observation can be interpreted three ways. Maybe the original sample is all asparagine, right? Show the left. So then the observation is artifact. Or to the right, the initial sample, authentic sample, has a little bit diamidation aspartic acid, but during analysis has much more. The only thing is this analytical result truly reflect the sample is there's no diamidation during sample analysis. So again, it shows a little bit of kinetics. So if you look at this column, if the half time is 1 day, shows here, and even half an hour it causes 1% diamidation. If the some diamidation half-life is 10 days, shows over here, and if 10 hours, it will be about 2%. If overnight 24 hours, it will be 6% diamidation. Again, this is during sample analysis. That will be artifacts. So you can see that this is a result from Jim Wilson's paper. Again, the condition is 37 overnight and 90 degrees for 10 days. You can see some of the events and the NS shows in this box. There's about 4% deamidation, and the NH shows about 7% deamidation on this column. But as I said, these NH and N as deamidation half-life is about 10 days, overnight condition will generate 2 to 6% deamidation just during the analytical process. So we can see that the results observed mostly is due to deamidation artifacts during analysis. So this calls to closer examination of the other deamidation they observed in this paper. So the good news is we develop a way to minimize deamidation artifacts during analysis. Because deamidation is pH dependent, and at around mildly acidic condition around pH 5, deamidation is slower. So we use a very common protease GluC and digest protein and peptides and pH 4.5 instead of 8. And my student Shanshan was able to see that we basically eliminated deamidation artifacts. So I'm going to the last process and also may cause some of the analytical artifacts. Characterization issue is on the bioconjugation. So antibody-drug conjugate become a quite a new modality, and this is an excellent work by Zhou at Amgen, and as he used the existing workflow to characterize many of the antibody modification sites. And this is another example of the challenge of detect all the chemical events for bioconjugation, and some of the events such as reversible modification of ADC has been difficult to detect. You have to use a different approach. So we also develop a workflow to look at bioconjugation and characterization method, and it shows over here we use a brominate fluorogenic probe, so we're able to detect all the bioconjugation product by fluorescent and the mass spec. So what we found that is using the typical workflow, we're able to detect a certain modification events, but there's many more the standard workflow missed, right? So we're able to use isotope and fluorescent to see that. One example is actually a side reaction of acylation reaction normally won't detect by the typical workflow. So this calls for we really need to be alert of the analytical process and analytical artifacts. So I give a quick view of lots of the topics my lab is interested in. Again, I'll be happy to talk to you offline about the details and also some of the new programs due to COVID-19 I'm not able to share with you. Finally, I just want to mention a program at Northeastern University. We call the Industry PhD program. A scientist in the company can remain as an employee at the company at the same time obtain a PhD with Northeastern University. We are really experiencing this program, and here's many companies currently or the past in the program. And many of the work I mentioned today actually was done by these students, such as Chris Chomsky, formerly at IV, now at BMS. With that, I want to thank you for your attention. Again, I'll be happy to share with you my slides and some extra slides. I'll be happy to talk to you about some more details and other ongoing programs in my laboratory. Stay safe and healthy.